Hello class, this videotape is for cell division mitosis. Mitosis, the mechanism of cell division is called mitosis. New cells only come from pre-existing cells, and then only cell division produce new cells. Two cells formed from mitosis are called daughter cells, and the cell before committing mitosis is called mother cell. So one mother cell becomes two daughter cells. In unicellular organism, that single cell is entire body. We call that unicellular organism. When this unicellular organism undergo mitosis, it forms two daughter cells. Since single cell is its entire body, that cell division is equivalent to reproduction. Reproduction means making identical organism. For example, when you have a baby, that is reproduction. But mitosis to human, you have a gazillion number of cells to form your body, and one cell undergo mitosis to produce two cells. That does not mean reproduction. But in case of a single cellular organism, unicellular organism, the entire body is a single cell, and then when this single cell undergo mitosis, that means reproduction. Today, you will only think about mitosis. In terms of reproduction, there's another kind of cell division called meiosis. Meiosis will not be covered today. Cells always give rise to cells that are like themselves. The goal of mitosis, the cell division is required for growth, development, and repair. Think about yourself when you are young, you were small. Now you've grown up and you're bigger. So the number of cells are increased by way of mitosis. That means of growth. Then repair, when you get injury or when you have a scar from accident, maybe when a small part of a tissue you lost, those cells can be generated by repair. At the time also, mitosis takes place. One of the significance of mitosis lies in that mitosis generating two identical daughter cells. Otherwise, it is impossible to divide the gene evenly. In other words, the copies of chromosome are passed from mother cell to daughter cell so that daughter cell receive a copy of mother cell's genetic material. Therefore, the daughter cell nuclei receive the same number and same kind of chromosome as the mother cell has. If some cell from your body has a different DNA integrity from some other cell of your body, this can cause a problem. You will study that at the end of this lecture. And where in your body does mitosis take place? Non-reproductive cell or somatic cell. Non-reproductive cells. So there's other types of cell called reproductive cell. As I said, reproduction is different. Reproductive cell, they undergo different types of cell division, which is called meiosis. Except those reproductive cells, the rest part, we call them non-reproductive cell or somatic cell, they undergo mitosis. Some category of cells keep dividing. Some other category of cells is permanently differentiated. So in case of some cell permanently differentiated, then they have mitotic replacement rate. In case of skin cell, it's about 2 or 3 weeks takes. Replace old skin cell to new skin cell. In case of stomach, takes 2 or 3 days to replace new stomach cell, especially inner wall of stomach. Red blood cell it takes bone marrow produce new red blood cell with the interval of 120 days. So once red blood cell formed, it stays 120 days in a human body, and after 120 days, red blood cell disassembled. Then some component of a red blood cell can be recycled. Then bone marrow provide new red blood cell. So here's when stem cell divide one Doro cell. This Doro cell remains a stem cell. So this stem cell behave like this stem cell. Thus, continuing the line, this line continued. Okay? And the other Doro cell eventually differentiated into blood cell, bone cell, muscle cell, or nerve cell. Some cells behave like a stem cell. Some other cells capable of dividing and permanently differentiate into cell. Therefore, this blood cell has some lifespan, such as 120 days. The stomach cell had 1 or 2 or 3 days, and skin has 2 weeks. Those are the replacement rate. When we study cell division mitosis, we really need to pay attention to chromosome and DNA, chromosome behavior. And then chromosome is composed of DNA. DNA is contained in a number of linear chromosomes. So we need to study the structure of chromosome first. DNA strands are coiled and twisted, so it can fit into a very smaller space, such as inside the nucleus. Chromosomes are composed of chromatin, and chromatin is composed of 40% of DNA and 60% of protein. So let's think about the size of a level. The chromosome is big, but the smaller level is chromatin. So chromatin make up of chromosome. But this chromatin is composed of 40% of DNA plus 60% of protein. So we think that chromosome is entirely composed of DNA, but that's not true. There's some protein, and DNA formed complex with the protein. The name of this protein is histone protein. And DNA strand wrap outside the histone protein. We call that nucleosome. Nucleosome look like bees on a string. Let's look at figure. Look at here. This is a DNA strand. Can you see green string? These are DNA strands. Can you see this blue like ball shape on the top 4, the below 4? So 8 histone. This is the name of histone. 8 histone protein formed this one, and then outside 8 histone protein, DNA wrap this 8 histone protein. We call that nucleosome. So this is nucleosome. This is nucleosome, and then this is nucleosome. So nucleosome look like bees on a string. These look like a bead, and then here is the below of more 4 bees, and then DNA is gonna wrap outside 8 histone octamer. Octamer means 8, and then we call these bees on a string. And this structure is called nucleosome. Many nucleosomes are packaged into thin string-like fiber, which is called chromatin. This is called chromatin. Nucleosome is composed of DNA wrapped outside histone protein. We call them nucleosome. Many nucleosomes, they package into thin string-like fiber. Thin string-like fiber, we call that chromatin. These chromatin undergo further condensation to form chromosome. The complex DNA and histone molecule coiled tightly form a compact chromosome, so the chromosome can fit inside the nucleus. Look at here one more time. DNA double helix. And then this is histone protein. When a histone protein formed, we call that histone octamer. And then outside a histone protein, this blue line indicate DNA strand. We call this nucleosome. Many nucleosomes packed together to form string like fiber. We call this chromatin. This chromatin further coiled and twist and compact to form chromosome. And then these chromosomes, they really tightly packed. Therefore, they can sit in small space such as nucleus. Let's briefly review about DNA structure. DNA, this is one strand of DNA. And DNA is composed of phosphate, sugar, and base. Sugar phosphate forms backbone of one DNA strand, and nitrogenous base protrude out of a single DNA strand, form complementary base pairing with other base protrude from the other DNA strand. So DNA always exists double helix. Sometimes DNA exists as a single strand very for short moment for specific other reason. Normally, DNA always forms double helix. In terms of sugar, there are two kinds of sugar. For DNA, this deoxyribose sugar. In terms of RNA, this is ribose. If you look at the second carbon, DNA does not have oxygen, oxy, but deoxy means no oxygen. Pentose sugar for DNA has this chemical structure, but RNA has oxygen, so they do not put deoxy, they just put ribose. There are four kinds of bases: adenine, guanine, cytosine, thymine. But they have a specific rule to make complementary pairing. So adenine always bind to thymine. Guanine always bind to cytosine. Based on their structure, if we look at adenine and guanine, you can see more bulky ring structure here. So this adenine and guanine belong to purine group. And cytosine, they show less bulky structure. So cytosine and thymine belong to pyrimidine group. Chemical bond must form between purine and pyrimidine, not within purine, not within pyrimidine. Why? Why adenine always base pair with only thymine? Why adenine cannot base pair with cytosine or guanine? If you look at adenine and thymine, you can see this is hydrogen bond. This dot line, this is hydrogen bond. One hydrogen bond and two hydrogen bond. When you look at base pairing between cytosine and guanine, you can see 1, 2, 3 hydrogen bond. According to their chemical structure, adenine and thymine can form only 2 hydrogen bond, whereas cytosine and guanine can make 3 hydrogen bond. Therefore, adenine cannot bind to cytosine or adenine cannot bind to guanine because the number of formation of hydrogen bond is different between adenine and guanine or between adenine and cytosine. So adenine always bind to thymine, cytosine always bind to guanine. One more time, when you look at this is a single DNA strand. When you look at the orientation of a DNA strand, one end of a DNA strand we call 5' end. Why? Because the 5th carbon on sugar attached to phosphate. So 5' phosphate end or 5' end. And the other end of DNA strand is, we call that 3' end because hydroxide group attached to 3rd carbon of sugar molecule. So we call that 3' end. always DNA strand has one end is 5' and the other end is 3'. Here's a DNA double helix. You can see here phosphate group and sugar and base. These nucleotides carry guanine and form base pairing with cytosine with the other DNA strand. When you look at this strand is a sugar phosphate backbone. 1 out of 4 base is protrude and then they form complementary base pairing. This is the normal form of DNA double helix. 2 DNA strands twisted. But once you uncoil 2 DNA strands, this is 1 DNA strand and then this is the other DNA strand, and then you can see base pairing. So if I provide base sequence of 1 DNA strand, you can pull out base sequence of the other DNA strand. If I only give C, and then you will figure out what will be base of the other DNA strand. So with the information of base sequence of 1 DNA strand allow you to pull out the base sequence of the other DNA strand. Homologous chromosome. When you look at chromosome inside the cell, you will find that 2 of each type of chromosome are identical. This is called homologous chromosome or homolog. According to a pair of homologous chromosome, the living organism carry different number of chromosomes. In case of Homo sapiens, this is human, how many chromosomes do you have? Please know about this. This is a little bit fundamental information. We have 46 chromosomes. What do you mean 46 chromosomes of human? That means human has many cells, and inside the cell, inside the nucleus, there are 46 chromosomes exist. In case of a potato, they have 48 chromosomes in individual cell. Also, tiger has 38 chromosomes in individual cell of a tiger. So each species has their own unique chromosome number. But when you look at the chromosome number, they all show even number. Even number can, a scientific denotation can be 2n. When n equal 23, then 2n you can write down, you just plug in 23 to n. So 2 double 23 equal 46. This is the chromosome number human cell has. If n equal 24, then 2n equal 2 multiply 24 equal 48. This is chromosome number of a potato. If n equal 19, 2n equal 38. So this is the chromosome number of tiger. So by using these 2n, the scientific notification, we can convert a variety of chromosome number through many different kinds of species. So 2n is called diploid. This 2n is a scientific notification and represent diploid. Why chromosome number always turn out to be even number? Because of homologous chromosome. When you look at chromosome inside the cell, you will see 2 of each type of chromosome present. We call them homologous chromosome. Because of homologous chromosome, the chromosome number of any species turn out to be even number. Human has 46 chromosomes. These 46 chromosomes also can be paired as homologous by comparing size, shape, location of a centromere. This arrangement of a chromosome is called karyotype. This is karyotype of 46 human chromosome. Okay. So both are homologous chromosome. This homologous chromosome. This homologous chromosome. Two chromosomes identical. And then scientists put them side by side and then put the number. Number 1 has two homologous chromosomes. Two chromosomes, they are identical each other. Number 2, there are two chromosomes, they are identical each other. So this is another set of homologous chromosomes. In this way, you can see 22 pairs of homologous chromosomes. The rest 2 are sex chromosomes. In case of a female, even XX, this sex chromosome also show homologous chromosome, but in case of male, they have XY. So in case of male, they have 22 pairs of homologous chromosome. So this is inside the cell. Do not look at square yet, just to look at this, the right side. When you look at blue color chromosome, can you see the other member of a homologous chromosome? Here. So scientists put side by side this way and then give a numbering. And this is a pair of homologous chromosome. You can do the rest of chromosome to make a pair of homologous chromosome. How you can make a pair? For example, look at here, this one and this chromosome. First, you have to see entire length of chromosome, whether entire length is similar. Okay, then look at this chromosome. Can you see not exact center, but a little bit a knob-like a structure? We call this centromere. Above centromere, we call this short arm. Below centromere, we call this long arm. And each chromosome has different lengths of short arm and long arm. And then look at the ratio between a short arm to long arm. And then if that ratio is same, then they are homologous chromosome. So look at this short arm, and long arm. Oh, both are pair of homologous chromosome. Then they put like here. So this is another set of homologous chromosome. So you can see this and this homologous chromosome. Therefore, in this way, human chromosome 46 chromosome is already analyzed which chromosome is a pair of a homologous chromosome. We call this karyotype. When you look at this set of homologous chromosome, these two chromosomes are identical. Today you might learn new terminology, chromatin, homologous chromosome, centromere. Now you are studying a sister chromatid. A sister chromatid means this word is related to cell division. When the cell decide undergo mitosis, think about it, one mother cell become two daughter cell. So this one mother cell increase content double so that they can evenly provide proper amount of cellular contents to each daughter. So before cell division, mother cell increase, doubling in terms of cellular contents as well as DNA content. So sister chromatid means before cell division, each chromosome replicate or duplicate, resulting in two identical copies. Sister chromatids remain tightly joined through centromere. Centromere is like knob-like structure in the middle of each chromosome. So this is one chromosome before cell division occur. This is unduplicated chromosome. It's not related to cell division. the cell does not undergo cell division every moment. Suppose one mother cell divides into two daughter cells, and then these two daughter cells sometimes stay long until the next round of cell division. Some cell immediately, the once daughter cell formed, then daughter cell sometimes quickly undergo another round of cell division. Every tissue, every cell has a different situation. But when you look at this unduplicated chromosome, this is not related to mitosis yet. But when the cell determines we need to divide, then this unduplicated chromosome in the mother cell increase amount doubled. We call this sister chromatid. When you look at these sister chromatid, do not count this chromosome as two chromosomes. This is wrong. Still one chromosome. Whenever you count chromosome, count the number of centromere. Even though the chromosome once duplicated, then centromere hold these two sister chromatid. Therefore, this is one centromere. Therefore, still this is one chromosome. But this one chromosome is duplicated condition. Chromosome duplicate increase DNA content in order to prepare for mitosis. We call that sister chromatid. Homologous chromosome is always there regardless of cell division occur or not. Okay. Sister chromatid only can be addressed in relation to mitosis. If the cell does not undergo mitosis, the chromosome doesn't have to be duplicated. Therefore, there is no sister chromatid. And then this is a centromere region. And then there is some protein complex called kinetocoa protein complex attached to centromere region. You will see this one a little bit later when cell undergo mitosis. Homologous chromosome, this is one member of homologous pair, the other member of a homologous pair. They are homologous chromosome. When the cell decide, okay, we are going to do mitosis, then one member of homologous chromosome duplicate, and the other member of homologous chromosome also duplicate. So they form sister chromatid. This is a sister chromatid. Still they are homologous pair, but this homologous chromosome in the state of sister chromatid. Without mitosis, homologous pair just is enough, it's okay without duplication. Non-duplicated form of a chromosome is okay if the cell is far from mitosis. But when the cell decide we need to undergo mitosis, then each member of a homologous pair duplicate, and then still they are homologous pair, but they form duplicated chromosome. We call this sister chromatid. And this one and this one, we call this non-sister chromatid. And inside the same chromosome, we call this sister chromatid. So this and each other, they are sister chromatid. But this chromatid, and then even though they are homologous pair, this one is called non-sister chromatid. Again, this is homologous pair. Here's a centromere. And for mitosis take place, this homologous chromosome duplicate or replicate a same meaning. And then they have sister chromatid. These are sister chromatid, two sister chromatid. And still they are homologous chromosome, but they form two sister chromatids. And this one chromatid and this, we call that non-sister chromatid. So do not confuse sister chromatid with homologous chromosome. homologous chromosome always present in the cell. A sister chromatid can be formed in relation to mitosis. Once mitosis is over, sister chromatid does not exist. Cell cycle, the division process itself, constitutes a complex cell cycle. The cell cycle consists of inner phase and mitotic phase. Mitosis, we call this mitotic phase. Mitotic phase immediately followed by cytokinesis. Except mitosis and cytokinesis, the rest phase is called inner phase. So cell cycle means each cell has lifespan with respect to mitosis. One round of mitosis occurs, and then until the next round of mitosis, we call that cell cycle. And cell cycle consists of inner phase and mitosis phase. You can include cytokinesis in mitosis together. It's not that big difference. Here's mitosis. And then the rest part, G1, S, G2, we call that inner phase. Inner phase is not a phase of mitosis. Inner phase set the stage for cell division. And if you look at inner phase also composed of 3 subphases, that is G1, S phase, and G3 phase. Entire cell cycle is composed of inner phase plus mitosis. And this inner phase is composed of G1, S, G2 phase. Therefore, if you look at cell cycle, 90% of cell cycle occupied by inner phase. What happened during inner phase? Before mitosis begins, the cell prepare. During G1 phase, cell's contents except chromosome are duplicated. So in the cytoplasm, maybe centrosome duplicate, endoplasmic reticulum duplicate, lysosome duplicate. Those subcellular organelles need to be duplicated. S phase, what happened? S means synthesis. Synthesis of what? Synthesis of chromosome or DNA. Can you see? Here is just a chromosome, but duplicated of chromosome. We call this what? Sister chromatid. Sister chromatids of each chromosome produced during S phase. Now, G2 phase, cell is very prudent. The cell double check the duplicated chromosome for mistake and making any needed repair. In the course of duplication of DNA, sometimes error can occur. We can call that mutation. So DNA mutation can occur, then that kinds of mistake can be fixed, can be repaired. If those mistakes is minor and then can be easily repaired, and then G2 phase proceed cell cycle to mitosis phase. Okay, so G1, S, G2 phase are required for preparing for mitosis. G1 phase, cellular contents doubled. S phase, chromosome doubled to form sister chromatid. G2 phase, just to check in the process of DNA duplication which occur during S phase. Is there any error occur? Is there any DNA mutation take place? Then G2 phase is going to fix those mistakes and make the cell enter mitosis phase safely. So cell cycle is composed of inner phase plus mitosis. Inner phase occupy about 90% of a cell cycle. And then you can read this, I just told you just before G1 phase, S phase, synthesis of what? DNA. G2 phase, just to checking one more time, before cell division occur, the cell monitor chromosome condensation, the synthesis of a microtubule, those things can be monitored before mitosis begins. So one more time, this is unduplicated chromosome. When cell does not undergo mitosis, chromosome has this configuration. But the cell decide to commit mitosis, the chromosome, the DNA doubled. We call this sister chromatid. So duplicated chromosome can be produced during S phase in cell cycle. And then you have to know the terminology of a centromere. At the center of a chromosome, there is a structure, we call that centromere. And then this centromere hold the duplicated sister chromatid tightly together. Even though this look like two chromosomes, you need to count the number of centromere. Therefore, this is one chromosome. Now again, here's centromere region. Outside the centromere, there's a protein complex called kinetochore attached to a centromere. On this kinetochore, later some kinds of a fiber, spindle microtubule attached to kinetochore at centromere. Okay, once again, this is unduplicated chromosome. When cell does not do anything for mitosis, the chromosome shows this unduplicated chromosome. But when cell decide mitosis, this chromosome doubled. But still this is one chromosome. Still one chromosome. They said one chromosome. And then the center part we call centromere. Just in case, do you remember there's some protein complex attached to centromere that is named kinetochore. Then when you see this different portion of color, this indicates gene. So one more time, here's entire cell cycle, and this is M phase, mitosis phase. Cell division occur. The rest part is called what? Interphase. Interphase also categorized G1, S, G2 phase. During G1 phase, the cellular content increase. S phase, DNA synthesized to form sister chromatid. G2 phase, they check cell stability and then they check about the preparation condition for cell division. And then mitosis also can subdivide into prophase, metaphase, anaphase, and telophase. We need to think about how chromosome behavior differently from prophase, metaphase, anaphase, and telophase during mitosis phase. Which of the following does not describe a sister chromatid? Sister chromatids have separate centromere. I said centromere hold two sister chromatids tightly. Still, we count that as one chromosome, one duplicated chromosome. Sister chromatid cannot have separate centromere. So A is wrong. Sister chromatids consist of double-stranded DNA helix. Correct. Each sister chromatid holds double-stranded DNA helix. Sister chromatids are part of a duplicated chromosome. Correct. And sister chromatids become independent chromosome after they separate. That's true also. This is sister chromatid. And then after cell division, these two sister chromatids actually cleave. One sister chromatid move to this direction, the other sister chromatid move to the other direction, and this is going to be one daughter cell later, and then this is going to be another daughter cell later. Let's look at mitosis phase. With C phase means cytokinesis. So M phase means mitosis phase. And mitosis phase also subcategorize prophase, metaphase, anaphase, and telophase. And after telophase, immediately cytokinesis phase follow. And then before we start prophase, metaphase, anaphase, telophase, let's study mitotic spindle first. Do you remember centrosome? The centrosome can be found only in animal cell. And then do you remember like this? Centrosome have this, a pair of centriole, and another like this. This is a centriole. A pair of centriole form centrosome. This is a centrosome. Upon mitosis, this centrosome produce spindle fiber. we call this spindle microtubule or spindle fiber. It's like a string structure. Microtubule is one of cytoskeleton. Do you remember? A microfilament, microtubule, intermediate filament, those three pertaining to cytoskeleton. And this microtubule produce fiber or string structure. We call that spindle fiber or spindle microtubule. These spindle microtubule only appear during mitosis. And the origin of formation of microtubule is from centrosome. And then two centrosomes are formed during interphase. So during interphase, actually, this single cell has this one set of a centrosome, but when the cell decides to mitosis, and then this centrosome duplicate. So two centrosomes are formed during interphase. Before mitosis begins, we call them interphase. And then each centrosome migrate to opposite end of the cell during prophase. So for cell division, another centrosome produce, and then this centrosome move to one end of cell. Imagine that another centrosome move to the other end of the cell. For each centrosome, microtubule extend out, and the spindle microtubule can be formed from this centrosome, microtubule, and aster. So during prophase is the phase within mitosis phase. Mitotic spindle or spindle microtubule formed during prophase here. So prophase is already mitosis begin. And kinetochore appear again, protein complex associated with centromere. Don't confuse of centromere and centrosome and centriole. The biologist put the word, this is a cell, and one end of a cell is pole. This like Earth, so maybe this is North Pole, and then the other end of a cell, South Pole, and then the center part is equatorial plane or equator. So the centrosome replicated during interphase. So before mitosis phase occur, centrosome duplicate, forming two centrosomes. Then one centrosome migrate to this opposite end of the cell during prophase, and the other centrosome migrate to the other end of cell during prophase. And from centrosome, there's a very short microtubule extend. We call this aster, look like a star. This aster is kind of a radial array of a short microtubule. This aster extends from centrosome. We call that spindle microtubule, extend out of centrosome. During prophase, this spindle microtubule attach to kinetochore of centromere. So here is centromere region. This is centromere. Outside the centromere, you can see some protein complex associated. We call this protein complex as kinetochore. This is a centrosome, is this centrosome. Chromosomes overlap, so it's not clear to find the centromere. But this microtubule extend from centrosome and attach to kinetochore. Chromosome align at equatorial plane. This is the most phenomena occur during prophase. So one more time, this area is centromere. This part is centromere. And centromere is part of a chromosome. Now, there is some protein complex called kinetochore. Spindle microtubule extended from centrosome, attached to kinetochore, and then pull one sister chromatid toward their pole. These prophase, metaphase, anaphase, telophase, they are part of mitosis phase. And before mitosis is what? Interphase. Interphase has G1S and G2 phase. So just at the end of G2 interphase, the prophase of mitosis occur. During mitosis, and then each phase of prophase, metaphase, anaphase, and telophase, you have to observe how chromosomes behave differently along each phase. Let's look at the chromosomal behavior first. In interphase, you can see nucleolus inside the nucleus. And then you can see clear nuclear envelope. And then you can see the chromatin is relaxed, uncondensed state, relaxed. So it's a little bit disentangled. And then relaxed, uncoiled DNA structure. Once mitosis begin, once prophase begin, then chromosome start to condense. You see chromosome become shortened, and then you can see spindle microtubule begin to form from centrosome. Before mitosis begin, you see only centrosome duplicate. Now, once mitosis begin, this centrosome move to each pore, and also spindle microtubule start to produce. A little bit later, so pro-metaphase is a late prophase. You can put this just into a prophase together, it's okay. During late prophase, what happens is this nuclear envelope dismantle. And the nucleolus, you cannot see during prophase. Nucleolus, this one disappear. What else? Even late prophase, you can see spindle microtubule extend more and more, and then try to attach it to kinetochore. This is the happening during prophase. This spindle microtubule attached to kinetochore during prophase. Then kinetochore microtubule lined up the chromosome at the cell's equator. The chromosome aligned at the center of the cell. In anaphase, this spindle microtubule attached to kinetochore and then pull one sister chromatid toward their pore. There are two sister chromatids. Two sister chromatids separate. One sister chromatid drag toward one pore, and the other sister chromatid drag toward the other pore by spindle microtubule. That is anaphase. Telophase. These set of sister chromatids all move towards close to each end of the cell, and one set of chromosome reaches each pore and begins to decondense. Nuclear envelope restarts to form, and nucleolus reappear. And spindle microtubule disappear, and there's some microfilament, another type of cytoskeleton, microfilament form ring here around the equator. So telophase is look like still one cell, but this cell has two nuclei. That is the state of telophase. Now, cytokinesis means these cytoplasm completely separate to form two independent daughter cells. So cytokinesis immediately occur right after telophase. So here's still one cell, and then there's two nuclei, and then at the center, there's microfilament formed ring. There's some ring. This ring is called contractile ring of a microfilament. This microfilament constrict toward the center more and more. So just pulling this ring inward so that the cytoplasm eventually cleaved. So now you see daughter cell. This is the cytokinesis of animal cell. The plant cell undergoes cytokinesis a little bit differently. So if we look at this plant cell, there are some vesicles aligned at the center first. And then these vesicles formed and fused together that vesicle formation move from center to outward. Cell plate forms, and then this cell plate become new cell wall. M phase means mitosis phase. Mitosis phase is composed of prophase, or prometaphase is late prophase, and metaphase, anaphase, telophase. You have to know the specific characteristic of each phase. Prophase, mitosis begin, and then what is important phenomena is nucleolus disappear, and two pairs of centrioles move toward opposite core of the cell. Spindle microtubule extend from centrosome, and also nuclear envelope dismantled, break down. One cystochromatid of each pair attached by spindle microtubule. That is the phenomena of prophase. What about metaphase? It's alignment of chromosome. The chromosome align in the center of the cell, equatorial plane, and that is metaphase. And the metaphase is the phase. We can see the rod structure of chromosome clearly. The chromosome structure shape highly condensed, and then it shows clear image on the microscope. That is metaphase. And anaphase is separate of two cystochromatid. So two cystochromatid separate, and spindle microtubule drag one cystochromatid to one pole, and spindle microtubule drag the other cystochromatid to the other pole. When each cystochromatid finally arrives at pole, each pole has one complete set of chromosomes. Then telophase is reformation of a nuclei. So it's still one cell, but there are two nuclei. Spindle microtubule disappear, and the nuclear envelope reform, and nucleolus start to appear again. And cytokinesis, cytoplasm divide, creating two daughter cells. And in case of cytokinesis in animal cell, they form cleavage furrow. So when this microfilament ring constrict toward inward, then this one, we call that cleavage furrow. And then cytoplasmic organelle have already been replicated and resorted to the area that will separate, become daughter cells. So cytoplasmic organelles have already been replicated during when? G1 phase of inner phase. Cytokinesis in plant cell plate form during cytokinesis. Vesicles align at the center first. These vesicles can be formed from budding up of Golgi apparatus. Golgi body has many extended vesicle condition, and then some of the terminal vesicle budding up from the Golgi apparatus and then align at the center of the cell. And then this vesicle is going to form together. So from center toward cell wall direction, this vesicle formation extend, and then eventually they form cell wall. So two daughter cells can be formed. Now, cell cycle control system, what is the meaning? The cell cycle, we already studied inner phase plus mitosis phase. That is a cell cycle. This cell cycle can be controlled. So the sequential event of cell cycles are directed by a distinct cell cycle control system. It's similar to clock. This clock has a specific checkpoint at which the cell cycle stops until go-ahead signal is received. So cell cycle control means there's some system going on in the cell to check each phase of a cell cycle. This cell cycle control system is regulated by internal and external control. So how this cell cycle can be controlled? There are three principal checkpoints. G1 checkpoint during G1 phase, G2 checkpoint during G2 phase, M checkpoint during mitosis phase. This is a mechanical analogy for the cell cycle control system. You can see this is inner phase, and the yellow sector represent mitosis phase. G1 phase, the cellular contents duplicate. S phase, DNA replicate. G2 phase, just check the condition of replicated DNA. But that is inner phase. But at the same time, there is some control system. There are 3 checkpoints. G1 checkpoint during G1 phase. G2 checkpoint during G2 phase. M checkpoint during M phase. What's going on these 3 checkpoints? From G1 checkpoint, there is some monitor. The G1 checkpoint ensures that the cell's DNA is okay for replication. If some cell can be damaged on DNA, and then depending on the degree of damage, some cell might be the G1 checkpoint say, okay, that degree of damage is okay. Why don't you go ahead to S phase? But if the DNA damage is too severe, then G1 checkpoint, wait a minute. Let's find how we can repair this damage. And then if G1 checkpoint cannot find repairment, then G1 checkpoint, wait a minute. Do not proceed to S phase. You'd better exit G1 phase and go to G0 phase, such as resting phase. In resting phase, the cell does not do anything. Just to stay, and then hopefully during that time, maybe fortunately, the cell can be repaired. But at least at G0 resting stage, is not part of cell cycle. It's an exit cell cycle. So G1 checkpoint determine the cell's DNA integrity. If DNA has not been highly damaged, then if DNA condition is healthy, then proceed, go ahead to S phase. And during S phase, a DNA replicate. And from G2 checkpoint, during DNA replication, there is some mistake can occur, some mutation can occur. Then G2 checkpoint monitor the mutation state of occur during S phase. If mutation occur too much during S phase, G2 checkpoint, okay, stop, no more proceeding. The cell cycle stop here, and then they do not undergo mitosis. So G1 checkpoint occur right after G2, right before the beginning of mitosis phase. Maybe the cell passed through, okay, the DNA integrity is good by G1 checkpoint, and DNA replicates successfully, so G2 checkpoint pass, and then you are committing mitosis. But even in the middle of mitosis phase, there is one more checkpoint. The cell monitor whether spindle microtubule attached kinetochore correctly, and then the cystochromatid properly pulls toward each pore. All these things check. And then if something wrong, one cystochromatid drag into wrong direction, then the chromosome number, the result of a daughter cell will have a different chromosome number. Then this M checkpoint stop. Even they do not proceed the rest of mitosis phase. So there are 3 checkpoints throughout cell cycle. G1 check, G2, M checkpoint. You have to know what is a specific check occur through this G3, G2, M checkpoint. Okay, so you read through again. I already explained with the figure, so you can read that. And then I said if the cell too much damages, then G1 checkpoint send the cell to G0 phase. This is a resting phase. So like this. If the cell pass through G1 checkpoint, then they can enter S phase. But if cell is too much damaged, if the cell cannot pass through G1 checkpoint, and then the cell should exit cell cycle and stay in G0 resting phase. G2 checkpoint, what happened? The DNA replication occurred. The second checkpoint is a G2 checkpoint, and then also they monitor whether DNA mutated a lot during S phase. And then M phase, M checkpoint, also they check this is the last checkpoint, and usually occur in metaphase from mitosis phase and trigger if something wrong, then they exit the mitosis phase and then they go back to G1 without a continuing mitosis phase. So this is the summary table. So you will see this is phase and mitosis constitute cell cycle. And phase detail divide into G1 phase, S phase, and G2 phase. And then M phase, mitosis, is gonna divide into prophase, metaphase, anaphase, and telophase. But this table emphasizes about cell cycle phase and checkpoint. During G1 phase, there is G1 checkpoint. What's going on? What does G1 checkpoint do? They check overall DNA integrity in the cell. Depending on the stability of DNA, the G1 checkpoint just give a signal, go ahead. So the cell cycle move to S phase. If the cell damage too much, the G1 checkpoint send the cell to G0, so quiet, the resting phase. Once a cell pass through G1 checkpoint, they undergo DNA replication, and during G2 phase, the second checkpoint called G2 checkpoint, they monitor whether DNA mutation occur during S phase or not. And then if the mutation level is not significant, and then G2 checkpoint has allowed the cell enter mitosis. Even during mitosis, there is the last checkpoint, especially metaphase checkpoint, whether chromosome behave properly. If something wrong, then even this metaphase checkpoint stop mitosis. Cell death means in tissue culture, and then cell line divide about 50 times. So it's petri dish. We put some cell, and then we can culture the cell culture. And then we provide food, nutrients. The cell has plenty of food, so this cell undergo mitosis. And then scientists are curious about how long these cells continue to do mitosis and how long these cells live as long as we provide food. And then they found that some cell line, they undergo mitosis 50 times. And then maybe it takes 1 month per mitosis. So 1 month means interval between next round of mitosis. So if this cell can divide 50 times, then that means this cell can live 15 months. Then scientists keep watching, keep observing, and then what happened after these cells undergo 50 times of mitosis, then these cells die, even though food continue to provide. So scientists curious about why they die. Who or what control some clocks, these kinds of a lifespan, a cell lifespan. This clock that keeps time by counting cell division. In other words, why they cannot continue more than 50 times? And if they have to die, if 50 times of cell division is the limit, then who control, who make this cell die? What kind of a program is there? Now, so they modify this cell culture a little bit. Okay, they do cell culture, and when this cell undergo mitosis 25 times, then they freeze this cell line. Freeze the cell under minus 100 Celsius for 1 year. 1 year. And then when cell is frozen, the cell is not die. When you thaw the cell carefully, the cell can be revived. So 1 year later, then they thaw this frozen cell and start to culture again. And then find that this cell start to over 50 times of cell division. And then the scientists found that once these cells thawed, then they undergo the rest 55 times of cell division, and then they eventually die. So already some programmed, the cell has already programmed to undergo certain number of mitosis, then die. Even though we provide plenty of food, this normal cell undergo certain number of mitosis and then they die. But cancer cell, they never ever die. As long as food supplies provide, then the cancer cell never ever die. They live forever, endlessly. So all other cells in your body, the normal cell, contain some kinds of clock. Okay, this cell line will undergo 50 times of cell division, mitosis, and die. Some other tissue, okay, you will undergo mitosis maybe 70 times, and then you die. Sometimes some cell, only you 20 times and die, something like that. This hidden clock is already programmed to the cell. This hidden clock keeps time by counting cell division, and when the last time of cell division comes and then the alarm goes off, the cell die. Then who control this hidden clock? What control this hidden clock? There's protein named growth factor. It's like a hormone-like molecule. This growth factor can be released when your tissue gets injury. Then the neighboring tissue, we need to help this damaged tissue, and then we need to speed up, we need to elevate mitosis so that this damaged scar or injured area can be replaced with a new cell quickly. So your body really has a good defense system. So a cell division cycle is regulated by sophisticated group of protein called growth factor. This growth factor can be released from neighboring cell when you get injury, when you lose some tissue of a cell, so that this growth factor is going to maintain homeostasis. So here's a growth factor release when you get injury. During development or when the cell get injury, a variety of cells release growth factor. And then this growth factor bind to growth factor receptor on plasma membrane. Once growth factor bind to growth factor receptor, then cyclin, here's another kind of protein, cyclin produced. And then cyclin, once synthesized, this cyclin then bind to cyclin-dependent kinase. They are all soluble form of a protein. They are not transmembrane protein, but in the cytosol, there's many kinds of soluble proteins are present. And one of them is a cyclin. Cyclin can be synthesized once growth factor binds to receptor on the membrane. Once the cyclin is synthesized, then cyclin binds to cyclin-dependent kinase, which is another protein fluorine in the cytosol. So cyclin and cyclin-dependent kinase complex is formed. These complex stimulate DNA replication. This cyclin and cyclin-dependent kinase complex stimulate the synthesis of a protein that are required for DNA synthesis. Therefore, the cell can enter S phase. And cyclin-dependent kinase complex not only stimulate certain protein synthesis, but also some chromosome condensation, nuclear membrane breakdown, spindle formation, attach of a chromosome to the spindle. All these kind of activity required during mitosis can be controlled by cyclin and cyclin-dependent kinase complex. This is the way how the cell can be controlled. So you can read this. This is kind of a review content. One of the important cell cycle regulator is P53. Just before we learn about cyclin with the cyclin-dependent kinase complex can control some protein which play a role during mitosis. But there's another protein named P53. This is a major player on G1 checkpoint. What does P53 do? What kind of monitoring occurs on a G1 checkpoint? Cell screen the stability of DNA before DNA synthesis takes place. If DNA is not mutated a lot, if DNA state is healthy, then P53 pass the cell, go into S phase. But if P53, this protein detect damaged DNA, then P53 halt cell division and stimulate the enzyme to repair the damage. So the cell start to repair the damage first. But if the damage level is high, then the cell might not repair that damaged cell. Then P53 determine, this cell cannot revive. It's impossible to make this cell healthy. Then P53 command the cell kill itself. We call this apoptosis, or committing suicide, or programmed cell death. So cell is really smart. If the cell is damaged too much, and then the repairing system of the cell cannot handle this damage, then P53 command, no, we'd better not keep this cell. Let's just make this cell die. We call that apoptosis, or committing cell suicide, or programmed cell death. Why P53 determined to kill the cell? Because P53 prevent the formation of a tumor. If this damaged cell continue to proceed S phase, continue to G2 phase and undergo mitosis, the daughter cells also highly damaged. The next generation granddaughter cells also damaged. And then the population of a damaged cell is propagate. That can form tumor. So before that happen, P53 control command, okay, this cell is too much damaged, too much mutated. There's no way to make this cell go back to healthy condition. Then, okay, this cell shouldn't be released, needs to go away. So that P53 does not want to have any tumorogenesis cell. But P53 is protein also produced by the gene P53 DNA. If DNA of 53 mutated, then protein p53 does not function properly. Then the damaged cell easily pass through G1 checkpoint. None of the protein bother this damaged cell at G1 checkpoint because p53 malfunction, cannot function properly. If p53 mutate, then it's a high probability to get into the process of cancerogenesis. In about 50% of cancer, p53 protein does not function because the DNA of p53 also damaged by maybe chemical or radiation. So the p53 protein no longer function properly. The abnormal p53 does not stop cell division, so the damaged DNA strand is replicated, resulting in damaged cell. So p53 has another name, is a tumor suppressor gene if p53 is not mutated. If p53 work normally, then this p53 prevent the origin of a tumor cell. Therefore, we call p53 tumor suppressor gene. Tumor suppressor gene protein trigger cell suicide, apoptosis. It's in the normal cell, but this normal cell mutate too much, excessive DNA damage. And then this cell cannot return to healthy state. Then p53, okay, this cell commit suicide. We call this apoptosis. And then this p53 monitoring just G1 checkpoint here at G1 checkpoint. So cancer is loss of cell cycle control, p53 mutated, and then G1 checkpoint does not function, and the damaged cell just go ahead through G1 as G2 M phase. So this damaged cell continue to propagate the another damaged cell, and they form colony. So the loss of cell cycle control is the beginning of cancerogenesis. Nucleus size of cancer cell is much larger than nucleus size of a normal cell. And then they have enormous capacity. They do not need even growth factor to stimulate, to produce some protein required for DNA synthesis. They have their own growth factor. Cancer cells have abnormal cell cycle control system. The cell cycle really shorter than normal cell. That's why cancer cells undergo mitosis much faster than normal cell, and then they form quickly colony. The normal cell converted to cancerous cell by a process called transformation. Okay? So cancer cells that are not eliminated by the immune system form tumor. The abnormal cells remain only at the original site. The lump is called benign tumor. Some abnormal cells just stay there. They grow. The number of abnormal cells grow, but they just stay in one spot. And then if a doctor find it early, then just from surgery, you just take out the lump of abnormal cell, then you'll be okay. Now, cancer, the growth disorder of a cell, the cell cycle control is disrupted. Unrestrained cell growth and division, and then a defect in the regulation of a cell cycle. Cancerous with multiple genomic problems, so DNA cannot be repaired, p53 mutated, not function properly, replication of a DNA, damaged DNA strand, and abnormal cells can proliferate. Then all these conditions brings cancer. You might hear about this tumor, benign tumor, malignant tumor, and metastasis. And tumor is a cluster of cells and then constantly expand in size because they have no regulation from cell cycle. So it occurs when the cell begins to grow in an uncontrolled way. So they live forever as long as they can obtain nutrients. Benign tumor, as I said before, is just the tumor cell surrounded by normal tissue. So we call that encapsulated. cluster of tumor cell just stay together, and then outside the tumor cell is normal tissue. So remove those tumor, then this benign tumor not spread to other part of the body, so no problem. But some malignant tumor can invade to other tissue. It's not encapsulated, invasive. So maybe the tumor started from muscle, but this tumor is going to move through circulatory system, maybe through blood, and then maybe this tumor can propagate in other region, in other place from primary site of origin. This is a malignant tumor. Even malignant tumor can be found early, then it can be removed, but still there's some risk still left over. If a malignant tumor has not been treated, then this malignant tumor progress into metastasis. This tumor can grow even far distant site from the primary site of origin of tumor. And then this metastasis occur, then even doctor cannot, even though some cluster of a tumor removed, still single tumor cell, we cannot get rid of it even at the cellular level because a single cell is very small. So after surgery, maybe the patient might feel a little bit better, but in the meantime, this tumor cell divide, undergo mitosis, and then another tumor grow, and then once tumor achieve the property of invasiveness, then that tumor learn and have memory, so they know how to invade other tissue. Therefore, it's metastasis can come back again. So cancer progression does not occur as single step. It requires several mutations. For example, mutation inactivate tumor suppressor gene, such as P53, and the damaged cell proliferate, and another mutation inactivate the DNA repair gene, another mutation of a proto-oncogene to oncogene, another mutation inactivate some other kinds of tumor suppressor gene. P53 is most representative of tumor suppressor gene, but there are some other kinds of tumor suppressor gene also present in the cell. And those also mutated, then through this multiple step, the cancer really develops. So cancer can be caused by series of mutation. So carcinogenesis is also normal cell, but through the series of mutation, then eventually this normal cell transform into malignant cell. And then tumor cell grow faster than normal cell. So usually they occupy larger region in human body than normal cell. And depending on the location of cancer, there is some different name of a cancer. Scientists put that. Carcinoma is most common type of cancer in areas. Carcinoma arises from the cell that cover internal body surface or external body surface, such as lung, breast, colon, most frequent cancer in carcinoma. And sarcoma are cancer region arising from the cell found in the supporting tissue of the body, such as bone, cartilage, fat, connective tissue. And that area, when tumor grow, the cancer arise, then we call that sarcoma. And lymphoma are cancers that arise in lymph node or lymphatic system. And leukemia are cancers of immature blood cells that grow in the bone marrow and tend to accumulate in large numbers in the bloodstream. We call that leukemia. There are 3 highest risk of cancer in the US. That is breast cancer and lung cancer and colon cancer. This record is about a couple of years ago. I'm not sure whether last year changed, but as far as breast cancer, Lung cancer and colon cancer are the more prevalent cancer in the US. In different countries has a different high top risk of cancer in different region. So depending on their lifestyle. Okay, this is the end of mitosis. Okay, let's solve the questions. Question 1. How many daughter cells are produced by mitosis? Mitosis begins with one mother cell, and once this mother cell undergoes mitosis, how many daughter cells can be produced? You can find the answer from the lecture. Second question. Which of the following is not one of three deadliest cancers in the US? You hear this from the lecture, so you can easily find the answer. So, this is the exact same pattern to submit assignment which you've done last week. First, this is email main text body. So you write down your last name, comma, followed by your first name. You write down answer of question 1 of ABCD, also answer of question 2. And this is my email address, and please include your email address also. And please submit this assignment until next Wednesday midnight. This is the end of lecture mitosis. Thank you.